All right, guys, welcome to 2.3. This particular section, we're going to look at organelle structure and function. Let's jump into it. All right, so organelles are subcellular structures, and each of them have a particular function. The ones that are highlighted are the ones that we're going to cover in this particular chapter. Here they are laid out for the plants, and then also here they are laid out for the animals, okay? Here they are laid out again in list form with some general structure and function for them. I'm just going to kind of give you guys a brief overview here, and then we're going to go through each one carefully in excruciating detail, so you might as well buckle down, okay? All right, so we start with the nucleus, then we're going to talk about the endoplasmic reticulum, the smooth and the rough. We're going to talk about the Golgi apparatus, mitochondria, and chloroplast, lysosome, vacuoles, and also the cell wall. The plasma membrane, which is in actually 2.4, the ribosomes and the cilia and flagella. Okay, let's jump into them. All right, we're going to start with the nucleus. So the nucleus controls the function of the cells, right? That's the nucleus right here. It is pretty much the defining feature of eukaryotic cells, and it is spherical and is usually enclosed in a double membrane called a nuclear envelope, right? The nuclear envelope contains protein-lined channels called nuclear pore complexes, or NPC, and those complexes are going to control the exchange of materials going in and out of the nucleus towards the cytoplasm, which we will look at why that is later. So the NPCs are made up of two materials. They are actually made up of ribonucleic acid, or RNA, and they are made up of proteins, which include ribosomal proteins. The cytoplasm is actually the rest of the cell, so that's different from the nucleus. So here's a nucleus under a bit of a stain there for you guys to see in that particular cell. So there's your cell, and then there's your nucleus, okay? Now, functions of the nucleus. It controls all of the metabolic activities inside of the cell by regulating which genes are being expressed, right? Metabolism is the chemical reactions that occur within the cell. All of the chemical reactions and all of the stuff that's going on in the cell is controlled by the nucleus, right? The nucleus is also responsible for the initiation of gene expression, the reading and translating of genes. It is also the place where DNA replication during cell division occurs, which we will look at in chapter 3, and then also the site where repair of genetic material occurs, right? This genetic material is in the form of DNA, deoxyribonucleic acid, which stores biological information for the function of cells, right? So here's a diagram of how that kind of all looks, right? Your DNA gets read in the process called transcription, and then it's kind of like photocopied into a molecule called mRNA, and then that mRNA goes out into the rest of the cell to be used as instructions for those particular function of those organelles, which we will look at in a second. All right. Inside the nucleus is also another particular structure called the nucleolus, right? More of like this kind of region, and it contains ribosomal RNA, or rRNA, for the synthesis of the ribosomes, right, which is another organelle. It's a dense cluster of ribonucleic acid, and it doesn't have its own membrane. So, you know, here's your nucleus, and then here's the nucleolus region, right? So, sorry, there's your nucleus, and then there's a nucleolus region. Okay, let's talk about the next one, right? The next organelle is the ribosomes. The ribosomes are responsible for protein synthesis. They are your little factories that make protein within the cell. And protein are really important because everything is made out of protein. So these are the factories that make the proteins that make up everything, right? They're very small and they're present in large numbers within a cell. Most of them are made up of RNA, 2/3 about, and then 1/3 is made up of protein, right? There are kind of two locations in which you normally find ribosomes. They are either free-floating, kind of just in the cytoplasm, and if those ones that are free-floating, they're going to be producing proteins that are used inside of the cell, or they are attached to the endoplasmic reticulum, another organelle, which are then responsible for exporting it outside of the cell. So you think about the cell, you know, if you have free-floating ribosomes, they're going to make proteins that the cell's going to use for themselves, but then if it's attached to another organelle like the endoplasmic reticulum, it's going to be exported and used elsewhere, right? Ribosomes contain two components. You've got your small subunit and your large subunit, right? The small subunit binds to mRNA from the nucleus, much like a conveyor belt. The large subunit binds to the small subunit and it's going to act like a docking site for another type of molecule called a tRNA to assemble the amino acids. Okay, so mRNA and tRNA are sometimes confused with one another, but they are both different types of molecules responsible for different things. Okay, mRNA are the photocopies of DNA that is going to be used as instructions, right? tRNA, as the name suggests, transfers molecules and collect amino acids to then assemble the proteins, right? It looks a little bit something like this. Here's your ribosome, right? Here is the large subunit at the top, and here's the small unit down the bottom. This little photocopy mRNA just came from the nucleus, and then it's being sent to this particular protein factory like a conveyor belt. And then the small subunit binds to this mRNA along this conveyor belt here. The large subunit binds to that, and then will allow another molecule called a tRNA to come along and attach to these kind of little docking areas here, right? Here's how the process works of protein synthesis. This is how your body makes proteins to be used, well, by the cell, by the body, right? Number one, the mRNA is transported from the nucleus to the ribosome, where it attaches to the small subunit, like so, right, along here. The small subunit is going to move along the mRNA like a conveyor belt. So you see the legs walking on that small subunit. The large subunit attaches to the small subunit. So the large one's the one on top here, right? That's going to attach to the small subunit that's walking along the conveyor belt, and acts as a docking site for transfer RNA molecules. Transfer RNA molecules look like these little triangles. I mean, in real life, they don't look like triangles, but let's just use the animation, right? And basically, the bottom half of the tRNA attaches to the particular message that's there, and then the top half is going to attach to a specific amino acid, which forms your proteins, okay? The tRNA needs to match the mRNA sequence to the exact. So you've got a specific molecule here that's going to attach to a specific sequence right here, right? So you can see, for example, if the mRNA sequence is GAU, then the tRNA molecule here has CUA to match it, right? If it's AAA, it's going to have UUU to match that particular sequence as well. And then the top half matches a very specific type of amino acid, right? So that tRNA is going to collect that amino acid, and it's going to bind to this particular subunit according to the correct mRNA sequence, right? The large subunit then has an active site that binds the amino acids together to begin to form this polypeptide chain. So there is the polypeptide chain, and every time a new one comes along and attaches, it's going to bind that that molecule of amino acid there to that molecule of amino acid there, forming a polypeptide chain, which then gets folded into a protein, which your body then uses. And that's how your body makes protein molecules, right? All right, let's talk about the next one, right? So I made a mention of it last time, the endoplasmic reticulum. The endoplasmic reticulum is an interconnected layer of flattened channels called cisternae involved in transporting materials within cells, right? This is what the endoplasmic reticulum looks like, all these little folds, the little cisternae. And there are two types of endoplasmic reticulum. There is what we call the rough ER and the smooth ER, okay? Their name is derived from the presence or the absence of ribosomes, okay? If it is rough, it contains ribosomes on it. And those ribosomes that are attached to the outer surface means that endoplasmic reticulum, or the rough ER, is going to be processing proteins that the ribosomes have just produced, right? So the rough ER folds proteins into their correct shape. They're going to attach any sort of sugar onto those proteins to form glycoproteins, and then they're going to assemble these larger proteins, which will then later on either be exported out of the cell somewhere else, etc. If they don't have ribosomes, we call it smooth ER, right? And so smooth ER lack ribosomes, and because they lack ribosomes, they don't have anything to do with proteins. Instead, they are responsible for the synthesis or the making of lipids, right? Lipids are really important. They're another biological molecule that serves a very important function. In hormone-producing glands, it's going to synthesize steroids, hormones like testosterone, which are lipid-soluble. In liver cells, the smooth ER is really important in detoxifying certain drugs, converting them to a water-soluble form. which then your kidneys can get rid of. In liver cells, the smooth ER also stores glycogen and breaks it down into the glucose we need, right? So here's the rough ER with the little dots of ribosomes attached onto them, and then here's the smooth ER that lack those dots and responsible for lipid synthesis. Next up is the Golgi apparatus. The Golgi apparatus is a stack of cisternae, the folds, that are responsible for packing and transporting substances inside and out of the cell. It is basically the postal service of the cell. It is also called the Golgi body or the Golgi complex. And unlike the ER cisternae, these ones are not connected, right? And what they do is they form these little sacs called vesicles, which are like boxes that you send in and out of the cell, right? So basically, when proteins formed by the rough ER need to be exported, they are packaged into these things called transition vesicles. These transition vesicles attach themselves onto the Golgi body, and they move up each fold of cisterna, and from there it can either be sent to another organelle, like for example the lysosome, or it can be exported out of the cell in what we call a secretory vesicle, and then attaches to the plasma membrane and then releases the contents out into the wherever it needs to go, right? This process is called exocytosis, and the reverse can also be true. You can collect them from outside the cell, package them into vesicles, and then send them to the Golgi complex to receive them, and then used by the rest of the cell, and that's called endocytosis. We will look at both processes more in 2.4. The lysosome. Lysosomes are your body's waste management system. They are fluid-filled sacs that contain a really large number of digestive enzymes. Digestive enzymes are responsible for breaking things down. So these enzymes will break down carbohydrates, lipids, proteins, and nucleic acid, which is pretty much almost every part of a living thing, to be reused by the cell, right? Characteristically, they're very small. Here is a diagram, and that your lysosomes are that small. By comparison, there's your nucleus and your endoplasmic reticulum. There's your lysosome, right? And they are only active at a very low pH level, 4.8 on average, right? The function, right? So they digest an excess of materials and recycle them. And then also, if the cell's kind of going a bit nuts there, and it needs to kind of get rid of some of the old damaged organelles that it has, it can undergo a process called autophagy. Auto meaning self, phagia means to eat, and autophagia means to eat yourself. So it breaks down the cell's own non-functional organelles that are either too old or damaged. And also in some cells, they also digest bacteria that have invaded the body. So for example, your white blood cells will use lysosomes to break down anything that they eat, right? Here's a diagram of how that kind of happens, right? It kind of eats it, and then you can see this is a white blood cell, and that's a lysosome, and then that's the bacteria getting kind of like spun around and digested in the lysosome. Okay, on to the powerhouse of the cell, the mitochondria. Mitochondria are organelles that are responsible for the production of ATP, supplying energy to the cell. Okay, living things use energy in the form of ATP, right? ATP stands for adenosine triphosphate, which we'll look at in a second. Cells obtain ATP through the process of cellular respiration, which occurs in the mitochondria, right? So your mitochondria looks a little bit like these little beans that are kind of dotted around, and they have a whole bunch of little flaps called cristae, and then they've got the matrix, and then it even has its own little section of DNA called mDNA, or mitochondrial DNA. Okay, now the number of mitochondria in a cell is going to vary. In general, the more active the cell is, like for example a muscle cell or a brain cell, it's going to require more energy in the form of more ATP, and therefore that particular type of cell will require more mitochondria. Liver cells have about 2,000, right? Whereas things like mature blood cells have none because they don't need it. They're just a transport material. Here is a little diagram of that particular mitochondria present in the cell. So the purple stuff is the mitochondria. You can see they don't look exactly like beans, but that's kind of how we represent them in terms of diagrams, right? There's your mitochondria working. They're not worms, they're mitochondria, okay? Here's a micrograph of the mitochondria. There you go, long and skinny. That's cool. Okay, Let's talk about ATP. ATP is very important for understanding how your body uses energy, right? Adenosine triphosphate is an organic molecule. It's going to provide energy to drive important processes, chemical processes in cells, right? You can think of it like cash, right? It's a molecular unit of currency, and it is found in all life forms, from bacteria to animals to plants to fungi and so forth, right? And they drive processes such as muscular contraction, nerve impulses, which is, by the way, how you think, and also things like chemical synthesis and chemical reactions in the body. Here is a chemical structure of ATP, and if you are very much interested, you can see there is the adenosine molecule is this particular part here. There's an adenine molecule there, a sugar molecule there, and then it's got 3 phosphate tails. Okay, this is very important. When you put ATP in water, it breaks it down and it undergoes a process called ATP hydrolysis. This produces adenosine diphosphate, meaning 2 phosphate is referring to the phosphate molecules, and it releases 1 molecule of inorganic phosphate and some energy along with it. Okay, you chuck ATP in water, that last little tail, that little bond there, that's going to break off, and when it breaks off, it releases just a burst of energy. And cells are going to capitalize on that energy when it bursts to supply the needs of the cell. Okay, so that molecule ATP, this is produced in the mitochondria, right? Here is a diagram of how your body uses it to exchange materials in and out. That little last molecule provides just enough energy to change the shape of this protein in the plasma membrane, releases that particular sodium molecule outside of the cell. Okay. All right, so cellular respiration is a very complex process, okay? It occurs in your mitochondria, and I'm just going to explain kind of the fundamentals of it in this particular unit, and most likely you will look at it again in Unit 3 and 4 biology. At its heart, it's a chemical reaction. It's a chemical process where you break down glucose and oxygen to produce carbon dioxide, water, and ATP, okay? So it converts energy from the food that you and I eat into usable energy as ATP. Turns out, right, even though you eat that piece of steak, or you eat your potato chips, or you eat your sugar lolly or whatever, right, your body can't use those molecules because they're too big for energy in your cells. Instead, what it does is when it gets it down to the glucose level, it's going to break it down even more into allowing the cell to produce ATP, which is the cash currency that it uses, okay? Cellular respiration has 2 types. It can be anaerobic, which, as the name suggests, doesn't require any oxygen, or it can be aerobic, which requires oxygen, okay? And this is kind of generally what it looks like here, right? C6H12O6 is glucose, and then you need 6 molecules of oxygen to produce and break it down into 6 molecules of carbon dioxide, 6 molecules of water, and ATP, right? And that ATP is what the cells use, right? Now, there is a whole subsection of the different stages that occurs, and that is for you to have a look at if you are interested in it. But essentially, you will be covering this in Unit 3 in biology in Year 12, okay? Chloroplasts, the next organelle that we're going to look at. Chloroplasts are the famous molecule responsible for photosynthesis in plants and in bacteria and algae, but not in animals, okay? Photosynthesis is the ability to kind of capture energy from sunlight to produce glucose, and a typical plant cell will have 10 to 100 chloroplasts. That's what they look like, the little kind of round thingamabobbies. And here is another micrograph of a chloroplast looking there and there, okay? The chloroplasts, like the mitochondria, actually have their own DNA as well. Chloroplasts are comprised of a outer membrane, an inner membrane, and a thylakoid system, okay? So the thylakoid system is responsible for the light-dependent reaction of photosynthesis, right? And it's made up of a few different things, right? The thylakoid are flattened discs, and these discs contain specific enzymes that facilitate that process of photosynthesis. When you stack the thylakoids together, you get the grana, and a grana is encased in a skin called the lumen, right? And then between the grana are what we call stroma, which is this kind of fluid-filled spaces that fill the in-between of the chloroplast. So chloroplasts are responsible for photosynthesis, and like respiration, photosynthesis is a chemical process, right? Except that it is almost the reverse of respiration. It converts carbon dioxide and water to produce glucose and oxygen, right? And you need the light capturing pigment called chlorophyll in order to occur, right? Chlorophyll is what gives plants their green color. It's a green pigment. And when you think about photosynthesis, photosynthesis is what allows plants to produce and make their own food in the form of glucose from inorganic things like carbon dioxide and water, right? Oxygen, funny enough, is actually a byproduct. It's a waste product. When plants produce too much of it, they actually dump it and they get rid of it because they don't need it anymore, which is ironic because we use it, right? The glucose can then be used by the plant itself to then go through cellular respiration, and the plant will get energy from the ATP produced by the glucose as well. So plants actually use both cellular respiration and photosynthesis, whereas animals can only use cellular respiration. Okay, here's the chemical equation: 6 carbon dioxide molecules plus 6 water molecules with the addition of sunlight, absorbing sunlight in through the chloroplast. Will give you 1 molecule of sugar and 6 molecules of oxygen. Okay. Here is a molecule of chlorophyll. This is what, chemically speaking, that's what they look like. And that characteristic in green color that you see, that is actually because when the sun shines down on a leaf, right, the leaf will absorb all colors of the sunlight except for green, which is why leaves look green, because they reject green light, which is what our eyes perceive, right? It's not just green light, it's actually a few other things. So for example, when you see plants in the autumn, you can see and you'll notice that the leaves are red and orange and yellow as well. It turns out there are different types of chlorophyll pigments in a plant, and each different one will absorb a different type of color, right? So here's your wavelength of spectrum of visible light, and visible light is made up of all of these wavelengths, right? But chlorophyll pigments will absorb different types, right? So you can see the blues and up to the greens get absorbed really well by chlorophyll A, chlorophyll B, and carotenoids, which absorb your kind of brighter colors. And they appear green because they reject this green. So you see how in the green wavelength, nothing really gets absorbed, and also in the yellow, nothing gets absorbed as well. And then if you go into the reds, you start to see other molecules of chlorophyll, like chlorophyll F, taking in effect. Here is more detail, which once again, you will cover in excruciating detail when you get to year 12. Now, while we're on topic of mitochondria and chloroplasts, right, I want to talk about endosymbiosis, right? So endosymbiosis theory suggests that organelles like mitochondria and chloroplasts were actually once free-living bacteria, which were eaten and engulfed by larger eukaryotic cells. But instead of digesting and eating it, it was actually rather kept alive by the larger cell and had this kind of mutual relationship with one another that was beneficial for both parties, right? The theory was first proposed by Lynn Margulis in 1967, and the idea is the word endo meaning inside, and then symbiosis, sim is together, biosis is living. So endosymbiosis is one organism living inside of another organism. Now, how does that benefit both cells? Well, the eukaryotic cell, the thing that ate it, free energy from the chloroplast bacteria, right? And then the chloroplast bacteria also has a more efficient way of producing energy, and it gets protection from this larger cell, right? So in many ways, it's this beneficial relationship between the two that then becomes permanent and causes them to become things like a eukaryotic cell. Evidence for this come from the fact that both mitochondria and chloroplasts contain their own DNA, and the DNA is also in the shape of a plasmid, much like the bacterial DNA, suggesting that at one point in time they were actually bacteria, right? Both produce by a form of binary fission inside of the cell. So actually, when the cell divides, the mitochondria divide on their own, and so do the chloroplasts, which is similar to how bacteria divide as well, and they are of similar size to bacteria. Here is a diagram of kind of how that happens. So, a very long time ago, along comes a cell, creates these little folds in the plasma membrane, becomes more complex, and then at some point, right, it eats the mitochondrial bacteria, and then it keeps it around, right? And this is kind of the precursor to the ancestral animal, right? Heterotrophic organisms like animals. And then maybe at some point, they branch off again and they eat a chloroplast as well, and then the chloroplast along with the cell wall, then give the ancestral kind of plant organism as well. So then, you can see that's how branches of the plants and branches of the animal break off from these ancestral eukaryotic organisms. Okay, let's take a look at the next one, the cytoskeleton. Now, the cytoskeleton is not a single organelle, but it's actually a whole structural framework of the cell. It supports and it holds up the cell and allows organelles to move around within the cell, and it moves the chromosomes during cell division. Turns out, your organelles don't just float in the cell, but there's a whole bunch of scaffolding that occurs and framework that allows them to move around via these particular mechanisms, okay? There are three main components. You either have the microfilaments, the intermediate filaments, or the microtubules, right? Microfilaments are composed of small actin subunits and filaments, right? This little string there, that's an actin filament, and then it also contains what we call myosin proteins that transport molecules along these actin filaments, right? So here is a myosin protein. This is a protein that your body uses, and this is like a transport material that it's going to slide around, right? And this myosin automatically kind of chemically it walks along this actin filament, allowing that sliding action to move organelles up and down this particular conveyor belt, right? This actin-myosin complex is also responsible for actions such as muscle contraction, right? If you think about your muscle cells, right, muscle contracting occurs when the myosin pulls the actin filaments together, and that pulling contracts and pulls the muscle closer to one another, allowing it to contract and allowing us to pull things towards our body and to move around and walk and so forth, right? Which is why muscles can only contract, but they can't stretch, right? Other than the microfilaments, you have what we call intermediate filaments. These are larger than microfilaments, and they form a support system by scaffolding the organelle, right? So the actin and myosin combo are used to transport little materials along. The intermediate filaments are used to transport organelles and keep them in place, right? And your intermediate filaments, this is what they look like along here. They're a little bit larger, 8 to 10 nanometers, and they have these little kind of singular subunits there, and they're like giant cables, right? Larger than the intermediate filaments are the microtubules. Microtubules are the largest component, and they're organized by a particular organelle called a centrosome. The microtubule is responsible for moving chromosomes during cell division, right? So here is your centrosome, right? And your centrosome releases these microtubules, and they are very large molecules, about 25 nanometers, and that's what your chromosomes move in, right? Here is a diagram of a cell, and the green highlights all of the cytoskeleton within the cell, okay? The cilia and the flagella. The cilia are what we call hair-like projections that are on the surface of cells, and the flagella are tail-like projections at the end of cells, right? Both of them allow a cell to move or create movement, like beating of the cilia to push things along. They consist of microtubules, which are encased in extensions of the plasma membrane, right? So the cilia look like these little hair-like things on the cells, right? And here is an animation of that. That's an animation of your lungs and how your lungs remove mucus inside the lungs out. Here is a single-celled organism with a cilia, and every time that beats, it moves it through the water. And this is sperm cells. That tail is called the flagellum, which is singular for flagella. And that's how the sperm cells move, right? And the flagella has this kind of rotary function, and then one single beat of this flagella allows it to kind of swim through the water. So sperm is a good example. Bacteria also have flagellum. So here's another one that's there. And then some bacteria have multiple flagella, like lots and lots of them that allow them to move as well. Vacuoles. So the second last organelle for the day, the vacuoles are membrane-bound liquid-filled spaces that act as storage components, right? These are very different from plants these are different in plants and different in animal cells as well, right? In plants, the vacuole is a single large one, which is what we call a cell sap, right? So here's a plant, and that's your cell sap. It's huge, it's empty, and when it's when it fills up with water, it makes this plant very turgid and firm. Plants also don't have lysosomes, so the vacuole contains the enzymes and performs that function of the lysosome, right? Here's another plant cell, and there, once again, that's the vacuole filled with water. In animal cells, we have a lot smaller vacuoles, which are temporary, and a good example is your fat cells, your adipose cells, forming your adipose tissue, which is forming your fat, right? They have these very large central vacuole, which stores lipids and fats, which is what your body uses as an energy reserve when it needs it. So that's what it looks like there. All right, the last one is the cell wall. The cell wall is very specific to plants, bacteria, and fungi, and not in animals. A cell wall is an additional layer around the plasma membrane, and it gives strength and rigidity. I often think of the cell wall as much like a suit of armor, right? It makes it really strong and firm, but it restricts your movement. Cell walls are made up of material called cellulose, hemicellulose, and pectin, sorry. The trade-off is that you lose flexibility with strength, right? Cellulose looks like this, right? These kind of sheets of sugar molecules kind of stacked together in long sheets, there, and there, and it is really, really, really tough. In fact, so tough that animals often struggle to digest cellulose without the aid of something like bacteria, for example, right? Because we lack an enzyme called cellulase, which breaks down cellulose, right? And so you got your cellulose microfibrils, and then you've also got these little pectin things there as well. And so the cell wall kind of sits on top of the plasma membrane, and it gives that extra reinforcement. All right, well, that's it for 3.3, and I'll see you guys in 3.4.